Hey students, it's Mr. Lee Trudeau. Here's a quick recording of the lesson that we did today. This is Unit 6, Lesson 2, where the main thing I did was I went over the process of translation. So, hopefully if you missed anything, this will help clarify any questions that you might have had. But I started off today by reminding us of the central dogma, right? The central dogma, which describes the main information flow inside of a cell, where we have genes in the DNA, which provide instructions to make proteins. And the way that occurs is that DNA is transcribed into mRNA, and that mRNA is translated into amino acids, which then fold to make the protein. And so you might remember from last lesson, right, that in a eukaryotic cell and a prokaryotic cell, transcription occurs with the help of an enzyme called RNA polymerase, which creates that mRNA molecule. But then specifically in eukaryotes, like we see over here in this diagram, right, that mRNA molecule that's immediately made is actually a pre-mRNA molecule that needs to be processed by adding that 5' cap, the 3' tail, and splicing the introns, or in some cases, alternatively splicing exons. And once that has occurred, that final mRNA transcript moves out of the nucleus and into the cytoplasm, where it is then translated into amino acids. Now remember that in prokaryotes, because there's no nucleus, right, translation and transcription actually happen pretty much simultaneously, since there's no separation and no processing that has to happen after transcription has occurred. Okay, but more about translation. So translation, the major cellular component that helps with this process is called a ribosome. And a ribosome, I can see right here, consists of 2 subunits. And actually, if we were to look closer, these subunits, a ribosomal subunit, consists of RNA, particularly rRNA, ribosomal RNA, and some proteins that are kind of clumped together into these structures called subunits. And the mRNA molecule actually fits between the small and the large subunit and is kind of red, like a ribbon going through both subunits. Now, other things to note is that within the ribosome, there are 3 sites, 3 binding sites. There is what's known as the A site, the P site, and the E site. And I'm going to talk about what happens in each one of those using this figure over here, right? So as the mRNA gets connected to or attaches to the ribosomal subunits, and the ribosome begins to read the mRNA transcript, it requires another type of RNA molecule called tRNA in order to translate this mRNA transcript into amino acids. And so the way that this works is that these tRNA molecules, which look like this, contain a specific anticodon that matches up with the corresponding codon on the mRNA transcript. And each specific tRNA contains a specific amino acid that corresponds to the anticodon and codon. All right, and all of that is summarized in this genetic code table right here. All right, so you'll notice that these are describing all of the codons that could be found on the mRNA molecule and the specific amino acid that that codon requires. So, for example, an ACU in the mRNA would lead to a threonine amino acid. So how does it actually work? Well, so as the ribosome is attached to the mRNA and is moving down the mRNA, these tRNA molecules begin to interact with those A, P, and E sites. So first with the A site, so this actually goes from right to left. So a specific tRNA with an anticodon that corresponds with the codon on the mRNA. So this, for example, AUG, corresponds with the anticodon UAC, right? Because A pairs with U and G pairs with C. Right? That particular tRNA is going to bring the amino acid that's needed. Then that tRNA is going to shift to the P site, And as that occurs, the amino acid that's attached to that tRNA will then form a peptide bond in the P site with the growing polypeptide chain that exists in the P site. Right? Then the tRNA that was originally holding that polypeptide chain will then move to the E site and will be kicked out. All right, and this process will continue. So a new tRNA molecule will then be used to interpret the next 3 bases. Will enter the A site. That amino acid will form a peptide bond with the growing polypeptide chain, and then that first tRNA would be kicked out through the E site. All right, and that process will continue until the mRNA has been read. Okay, now translation, again, is resulting in this chain of amino acids, which then folds into a protein. Next week, we're going to talk about how mutations affect the process of transcription and translation and how it can result in different protein structures. But for right now, that's translation in a nutshell, and I hope that this cleared up anything. If you do have any questions, please reach out. Have a great weekend.